Today, it's my great honor to introduce our published paper in iMeta. The title is Differential Microbial Assembly Process and Co-occurrence Networks in the Soil Root Continuum along an Environmental Gradient. Soil harbor highly diverse microbial community with numerous functions and provide a seed bank for root-associated microbiota. The microbiota in the soil root continuum are influenced directly or indirectly by biotic and abiotic factors. However, systematic study considering the microbial community in 4 microhabitats along the soil root continuum across large geographic scales are still limited. To address this issue, we analyzed the bacterial community in the soil root continuum, surveyed at 82 field sites from warm-temperate grasslands to desert grasslands of the semi-arid and arid Loess Plateau, China. That is well known for its thick losses and severe soil erosion. For each sample site, we separated rhizosphere soil, rhizoplane soil, and also collected root sample of the dominant plant species and do the 16S amplicon sequences. Firstly, our results show that ANOVA analysis and PCLA results showed that microhabitats, grassland type, and plant species had significant effects on bacterial diversity, while microhabitats were the most important factors. And we also investigated the relationship between bacterial community along soil root continuum and the environmental factors. Without a surprise, the influence of environmental factors gradually declined from the soil and the rhizosphere to endosphere, and the mean annual precipitation was most important environmental factors to all parts of the bacterial along the soil root continuum. And then we found that environmental factors also affect the microbial recruitment in the soil root continuum. With decreasing mean annual precipitation, the soil root-associated bacterial community showed poor connection with the soil, but an increased connection with the endosphere bacterial community was observed. Non-model results show that there are more deterministic processes dominated than stochastic processes in rhizoplane, rhizosphere, and soil. With the increase of variable selection, while more stochastic processes dominated in endosphere, this is also consistent with the neutral model results. And last, we observed different modularity along the stress gradient in different parts, and we also found that the ratio of negative with positive cohesion decreased from high stress to low stress for root-associated microbial community, indicating the co-occurrence network of soil and root-associated microbial did not destabilize at a location with higher environmental stress. In summary, in this study, changes in microbial in the soil root continuum along an environmental gradient were investigated, and we found that the soil, rhizosphere, rhizoplane community were governed by deterministic process, and the environmental stress enlarged the dissimilarity of microbial communities in the soil root continuum. And last, we found that the root-associated microbial did not destabilize at locations with higher environmental stress. This is my presentation. More detailed information can be found in iMeta website. Thank you. And thank iMeta for publishing our work. Thanks.